a synergy webinar series around animal welfare. It is above all a synergy collaboration between three COST actions. COST actions are a European initiative to support networking in science and technology. And we are fortunate enough to have three actions where animal welfare plays a central role in each of them. I myself, I'm involved in all three, but I'm, well, in two of them really. I am above all active in the action IMPROVE, which is around the 3 R's in biomedical research. And we were also, this is also our initiative, very much because I felt and managed to convince some of people in the working group that I lead that we need to learn a lot from other people who work with animal welfare from perhaps a bit of a deeper science perspective than the refinement perspective that we take in biomedical research. The other two actions are LIFT, which is on positive animal welfare in farming. And I think LIFT and IMPROVE are both more or less on the same time schedule. So we're halfway through the life of the action. Whereas the third action, AFFECT-EVO, is more recent and started last year. And AFFECT-EVO is looking at the effective aspect not only of animal welfare, of emotions, the effective neuroscience across species. And I'm here just to give this general introduction today. I'm going to hand over to Rohish, who is the coordinator of the webinar series, and who will present today's speaker.
Thank you, Anna. So I'm delighted to welcome Dr. Tom Smulders, reader in evolutionary neuroscience at Newcastle University, with a PhD from Cornell and postdoctoral research at Duke and Wake Forest. Dr. Smulders is a leading expert in brain evolution and animal behavior. He currently serves as the action chair of the AFFECT-EVO, COST Action, and has led major international initiatives. A committed educator and an advocate for diversity in science, he also contributes to several editorial boards. He's fluent in Dutch, English, and French, which is very impressive. And outside academia, Tom enjoys ballroom dancing and is a fan of science fiction, a passion I'm pleased to share with him. Please join me in welcoming Dr. Tom Smulders. Dr. Tom, we are looking forward to your talk.
Thank you, Rohish. Always be careful what you put online about yourself because it might come back. Right, I will share. Oh, okay. I need to be able to share. So I've just sent a request to be able to share.
Yes, you can share now.
Okay. So I will share my screen. And just check with Rohish that you can actually see it.
We can see it.
Perfect. All right. First of all, I'd like to thank Rohish and Anna and everybody who's putting together this webinar series. First of all, for putting it together, because I think it'll hopefully be an interesting synergy among the three COST actions. And secondly, for asking me to be the first speaker in it. So I hope I live up to the expectations. I'm going to talk today about adult hippocampal neurogenesis as a potential valence marker and what it is. As I was putting this talk together, it really made me think more, as these talks always do. And so what I'm presenting to you is thoughts in progress rather than, this is my absolute conviction of how things work. I have to admit that when I started putting this together, I thought, well, maybe it is a valence marker. This is good. And by the end, I was like, well, maybe it isn't. So here's some other possibilities. So be prepared to not be convinced of a particular point of view, but rather to be presented with a number of potential points of view and some data to back them up from different perspectives. Okay, so on the first slide here, I'm showing you the funding support I've had over the last few years to work on this kind of aspect. And let me start by saying something that probably is not going to be a problem for most people, which is, why do we look at valence at all, and what does it have to do with animal welfare? And I'm sure nobody here is going to be surprised to hear that animal welfare as a concept, of course, has many different facets to it, many different aspects to it. When it's being assessed on farm, and it's being assessed in animal research facilities, a lot of different indicators go into it. Some of them are resource-based, you know, is something provided, is food there, is water there, is enrichment there, all these kinds of things. And some of it is animal-based. And when it comes to animal-based indicators, health is, of course, one of the components, but affective states are becoming a much and more important aspect of our assessment of animal welfare. So how is the animal feeling, if we want to put it in lay terms? And among affective states, one of the very dominant models about how to think about affective states is this dimensional model that suggests that all affective states can be mapped somewhere in a 2- or 3-dimensional space. I took one example here from a paper, but there's many versions of this. And this 2-dimensional space, the ones that are most often talked about, include a dimension that would be called arousal. Arousal in vertebrates and humans would be kind of activity of the sympathetic nervous system, activity of the HPA axis, things like, you know, increased heart rate, increased blood pressure. That would be arousal. In other species, it might very well be different physiological things, but that's the kind of aspects of arousal. Whereas valence, according to this kind of model, is orthogonal to arousal. And valence is about whether you experience something as pleasant versus too unpleasant, positive or negative. And so what I'm going to tell you about today is whether adult hippocampal neurogenesis might be an indicator of valence independent of arousal. But as you'll see later, actually arousal does come into play as well in how we think about what adult hippocampal neurogenesis might be indicating. Okay. Now, I've talked about adult hippocampal neurogenesis. Oh, sorry, yes. And as I go through talking about whether it's potentially an indicator of valence, I wanted you to keep in mind that in order for something to be a valid indicator or biomarker of something like valence, it should obviously have something quantitative that co-varies with valence. So that goes, you know, up when valence goes positive and down when valence goes negative, or vice versa. It doesn't matter which direction the correlation is in, but it should correlate in both directions, both for positive and for negative, and it should do it in a monotonic way. And secondly, and so part of that, so in order to co-vary, it shouldn't just co-vary with the valence of the situation, because the situation doesn't have valence. It's the experience of the animal that has a valence. So the same situation might be experienced by some animals as positive and others as negative. And if you have a valence marker, it should co-vary with the animal's experience of the situation rather than with the situation itself. And that's something we have to keep in mind. In addition, if you, in an ideal world, you find a gold standard indicator of valence, and then you see if your measure correlates with that gold standard of valence. But that's not an easy thing to do, because it is very hard to measure valence to begin with, especially in non-human animals. There might be a few things we can be fairly sure of are positive, some things that we're pretty sure are negative. And so we will be using those because there's very few other indicators to go by. But keep that in mind again, when I present evidence to you, critically think about, okay, so is this correlated with a gold standard indicator? Are we convinced that we're looking at something negative when we're saying, and therefore this is an indicator of negative valence? I just wanted to put those concepts up. All right, so that's about valence, which probably needed no introduction to everybody. Adult hippocampal neurogenesis may need a bit more introduction, because many people here, I'm assuming, are not neurobiologists. And so let me talk you through a little bit more what I'm talking about here. I'm going to start by talking about it in rats and mice, because they've been studied the most by far in this context. So we see here a picture of a rat or a mouse brain. Depending on the scale, they look fairly similar to each other. And in that kind of semi-transparent brain, we can see those two pink bilateral structures in there, which is what's called the hippocampus. So the hippocampus sits underneath the cortex in each of the two hemispheres. It's a fairly big structure in rat and mouse brains. Relative to brain size, it's quite large in rats and mice. Now, the first thing to know, and that many people may not be aware of, is that the hippocampus does not seem to be a unitary structure in terms of its function. It's become very clear over the last kind of 30 years that the septal pole, also sometimes called the dorsal hippocampus when it comes to rodents, or the rostral hippocampus, this area here, hold on, let me turn my laser pointer on. So this area here is very much involved in cognition, memory, maybe spatial navigation, depending on who you believe. Whereas the other end, we'd call it the temporal pole here, sometimes called the ventral hippocampus or the caudal hippocampus, is, due to its connectivity in the brain, much more connected to emotions and stress regulation than to cognition. And so even though internally the hippocampus looks fairly similar all the way along this long axis, in terms of its inputs and outputs, they are very different. And that sets up a different function for these different ends of the hippocampus. There's not an obvious boundary somewhere, so it seems to be a gradient from the one to the other along that axis. Okay, so now I'm going to explain hippocampal neurogenesis. So for that, we have to take a cross-section through this banana shape. And just as an analogy, imagine that you're taking a cross-section through a rolled up cake like this. So imagine this is your hippocampus, and we're now taking a cross-section through that. When you take a cross-section through the hippocampus, you are, just like in this cake, seeing this dense layer of cell bodies. Actually, you see two dense layers of cell bodies. So instead of the one sheet that's rolled up like in this cake, we have two sheets of cell bodies. One we call the Ammon's horn, and one we call the dentate gyrus. And the dentate gyrus is kind of folded around the tip of the Ammon's horn sheet. But this is a cross-section, of course. In 3D, this is basically a sheet, not a line. Yeah? So we're looking at the similar kind of sheet that's rolled up. Okay. Now, the dentate gyrus is where, in rats and mice, all the action takes place that we're going to talk about today, because that is where throughout adulthood, new neurons are being added to the hippocampus. The dentate gyrus is made up of a cell type called granule cells. They are very small cells, and there's many, many of them in this thick layer of cell bodies. And so what we have here, as we zoom in on this, is at the bottom of that thick cell layer, there are a set of stem cells. Stem cells that will basically bud off daughter cells, which will then multiply, and eventually some of those will mature into neurons. Others might mature into glial cells, but many of them will mature into neurons. And so that's happening here at the base of the dentate gyrus. And then once they mature, those cells migrate into the rest of the dentate gyrus and become like the other mature neurons in there. What we have is therefore different stages of development, and people measure the hippocampal neurogenesis using different markers, sometimes markers of cell division, sometimes markers of cellular maturation. And I'll show you some of both of those. Okay. So throughout life, new neurons are being added to the dentate gyrus. And when I talk about the rats and mice, that's what I'm talking about. Okay. So here is three of these markers. So we have, again, hopefully you see here the dentate gyrus, that kind of arrow-like shape in these 6 pictures. On the left, we have animals from a control condition, on the right from a chronic stress condition, and we have 3 different markers of neurogenesis. So Ki67 is a marker of mitotic cells. So it's an endogenous marker. It just means when we can stain for that protein, that means a cell that's expressing that protein is currently undergoing cell division. So mitosis, so therefore early stages of neurogenesis. And there's more of these in a control animal than a chronically stressed animal. BRDU, which we'll see coming back many times, is an exogenous marker. So you have to inject it into the animal, and then it will be incorporated into the DNA during the S phase of mitosis. And then we can stain for its presence. So, but it also means we can chase or follow neurons later on in life. So we can inject at one point but only kill the animal 4 weeks later. And then we can see which neurons, using a different marker to see that it's a neuron, were born during the time that the injection was made, because they will incorporate the BRDU in their DNA and keep it in their DNA for the rest of their lives. And so we know when that neuron last underwent a cell division, and that's when we injected the BRDU. And again, you use BRDU, you find fewer of these neurons in stressed animals than in control animals. And the last one here is double cortin. Double cortin is basically a protein that's involved in this part, in the maturing of the neurons. And it's a protein, it's a cytoskeletal protein that's involved in reshaping and making the growing out different processes, etc., in the neurons. And so you can see the processes, you can see the neurons expressing the double cortin, and again, there are fewer in stressed animals than in control animals. So just so you know that different markers basically tap into different stages of the neurogenesis process, but they're all tapping into that neurogenesis process. Okay. So that tells us negative experiences, chronic stress, reduce the number of these new neurons, no matter how you look at it. And so that's the inspiration for this might be a valence marker, because when animals undergo negative things, there's fewer of these that will survive, that will be generated, etc. And here, I want to get at the other end of the valence spectrum. So does positive things increase the number of these neurons? And first of all, so this study by Lehman from 2013, in their first experiment, basically just repeated what I just told you, that chronic stress reduces the number of new neurons. So we're looking at BRDU positive cells here. So that means BRDU was injected, and we see how many of those dividing cells there are in the dentate gyrus. We're looking at the rostral versus the caudal. Remember, the rostral or septal pole is the more cognitive one. The caudal or temporal pole is the more stress-related one. And then what we're seeing here is animals, look only at the black bars for the moment, that either lived in home cage conditions or had social defeat stress for 2 weeks. And we see that the number of BRDU neurons is lower in the animals that had social defeat stress. What this paper points out, though, is that if you remove the adrenal gland, and therefore its ability of the HPA axis to respond to stress, and you do that before the social defeat stress, the reduction doesn't happen. In fact, in their particular condition, it was an increase. So without the increase in corticosterone due to the chronic stress, you get a very different effect. So it means corticosterone is responsible, at least partially responsible, for this decrease. Okay. Now, what's interesting about their paper is they then did a follow-up experiment in which, after the home cage and social defeat, they gave the animals 3 weeks of enrichment. Because I didn't want to overcrowd the slide, they also had animals that just lived in home cages for 3 weeks here as a comparison. And we're seeing a similar graph here as we see here, where again, we look at the dark bars first in the caudal dentate gyrus. Now, this is home cage and social defeat originally, and it's then compared to, so this delta here is basically saying this is significantly higher than animals kept in a home cage. So in the sham operated animals that have their normal HPA axis, they have a higher number of new neurons when they've lived in an enrichment for 3 weeks than if they lived in the normal home cage for 3 weeks. But interestingly, if you take, again, the adrenal gland away, and therefore the ability of the HPA axis to respond, that increase doesn't happen. So you need corticosterone both to decrease in response to chronic stress, but also to increase in response to enrichment. Now, our traditional story would be that HPA axis is related to arousal, whereas neurogenesis here is responding, even though in both cases, corticosterone goes up. So if you measure corticosterone, as they did also in the paper, during enrichment, it is higher than in the home cage. If you measure it during social defeat, it is higher than in controls. In both cases, corticosterone goes up, but in one case, neurogenesis goes down, whereas in the other case, neurogenesis goes up. Positive experience of enrichment increases neurogenesis. Negative experience of social defeat decreases neurogenesis, suggesting that we might have a valence marker here. Unlike corticosterone, which goes up in both cases, which probably is an arousal marker. Okay, so both depend on corticosterone. Now, you could argue, and I've often argued, that enriching a cage for mice is very unlikely to actually be a positive experience. It's probably just relieving the normal negative experience of living in a mouse cage. And so there's an argument to be made that maybe we're just, making it less negative. We're not really making it in an absolute sense positive. And there aren't many studies, and this, people from the LIFT cost action will know what I'm talking about here. Positive affect hasn't been studied nearly as much as negative affect. But there's one study that I found which might come close to a gold standard positive experience in rats, that they then looked at what that did to neurogenesis in the hippocampus. And that's tickling. So rats apparently enjoy being tickled. Rats will come to a hand that tickles them. They will search out the tickling experience. At least some individuals will, some won't. And you can tell also whether they enjoy it because rats make high 50 kilohertz vocalizations in response to positive experiences and lower 22 kilohertz vocalizations in response to negative experiences. And some rats will do the high ones to tickling, someone will do the low ones to tickling, and the ones that do the low ones to tickling are also the ones that don't seek out the tickling, that run away from the hand, etc. So we have some good evidence here that, at least for some rats, this is a positive experience. And what this particular study did is it had a control group of rats and a tickled group of rats. It was only 5 days worth of tickling in this particular study. And all the rats responded with the 50 kilohertz calls. So they measured that. They made sure that it was positive for the individuals in the study. And what they found was that a month later, they then looked at how many new neurons, so new N is a marker of neurons, how many new neurons, but also just how many cell divisions there were. And it was increased in the tickled rats compared to the control animals, suggesting that something that is, probably the closest I could come to finding a gold standard positive experience for the rats does seem to increase neurogenesis in the hippocampus as well. So it isn't just about less negative, it might very well be about actually positive too. So as an interim summary about the rats and mice then, we have adult hippocampal neurogenesis going down with negative experiences, coming up with less negative experiences, so like enrichment, which includes, by the way, usually includes running wheels, and running wheels are known to increase neurogenesis. The running wheels are also known to be quite attractive to rats and mice. Mice will run them in them very spontaneously and for a long time, suggesting that, again, they are naturally motivated and enjoy doing that. But we also have this tickling experiment, which suggests that even when we go over and above neutral, if there is such a thing, that might also increase neurogenesis. So it looks like adult hippocampal neurogenesis in rats and mice follows the valence of the experiences the animals are having. I say that the time course is not fully clear because remember that most people have not studied this as a marker of valence. Most people have studied this to look for depressive-like states or something like that in the rats and the mice. And so they're not usually interested in how much of an experience do you need to have before the marker becomes visible, before you see a change. And so nobody's done that kind of systematic, how many days do you need of a certain experience in order to be able to pick it up with this marker in the field. Okay, so this all suggests to me we might have a valence marker here. And so that's rats and mice. There's a debate, Lucas here, about whether this kind of adult hippocampal neurogenesis is as important in all mammals. But what we do know, because there are other mammals that might have more plasticity in other parts of the brain, and we have no idea whether that responds to positive or negative experiences at all. There's big debate about whether humans have adult hippocampal neurogenesis and how important or relevant it is in our everyday functioning. there's two camps out there, ones that are convinced that it does, and ones that are convinced that it doesn't. So we'll have to see. But of course, mammals aren't the only animals out there. And the hippocampus is a very highly conserved structure across all vertebrates. So developmentally, it comes from the medial pallium, and there's a medial pallium in every developing vertebrate brain. And you can identify a hippocampal structure, a hippocampal-like structure in all amniotes for sure. So we have a bird here, a crocodilian, a sphenodon here, which is obviously one of those more primitive reptiles. But in fact, you can go to amphibians and find medial pallium. You can go to bony fish, find the medial pallium, although because of the development of their brain, the medial pallium actually ends up on the lateral side of the forebrain in them. But in all of these animals, it seems to be homologous, and it seems to have conserved some function. So it might be that what we find in rats and mice could be generalizable to many, many other species. And so in my lab, we worked on chickens quite a bit. I'm starting to realize that I have severely underestimated my how long I'm going to talk about this, as you can tell from the slides. I will speed up slightly here. Okay, so just to say that at least in birds, we think that same long axis between rostral and caudal, or between dorsal and ventral, between septal and temporal, exists there too. So we think that's also a conserved feature. And so in rats and mice, we were looking at the ventral hippocampus, and in birds, we're going to be mostly looking at this posterior hippocampus, which we think is homologous to that area of the rat and mouse hippocampus. Okay, so quickly then to take you through what we've done in birds. First question was whether chronic stress also reduces neurogenesis, and we used double cortin, remember that marker of plastic neurons, of maturing neurons, as our marker for to count in the hippocampus of chickens in this case. We did a chronic mild stress experiment, unpredictable chronic mild stress, where every day something different happened. The photo period was also very messed up. So there's a lot of stressful things happening to the animals for about 8 weeks. And to quickly cut to the chase, we found that especially in the caudal hippocampus, which we believe is the equivalent of the ventral hippocampus of rodents, we find a big difference. So we find a big suppression of new neurons compared to a control group that didn't have all those stressors over the last 8 weeks. To quickly orient you, in our lab, we've been routinely Z-scoring our measurements, and that's because there's a bit of variation from staining batch to staining batch. And so we basically Z-score, which means the mean is 0, the standard deviation is 1. So within a batch, we Z-score every time, and then we can combine the batches. And we, of course, make sure that a batch always contains equal numbers from each treatment group, randomly assigned to those batches, but equal numbers to each batch. So that's why you'll see negative numbers. In Z-scores, of course, because the mean is 0, you're going to get negative values as well as positive values. So that doesn't mean negative densities, it just means lower densities, just so everybody, because you're going to see a lot of these graphs that show this. Okay, so stress reduces these double cortin positive cells in the hippocampus, especially in the caudal hippocampus of chickens. That was an experimental stress manipulation. So then, I mean, that's all I've basically said here. So we then also looked in a more commercially and naturally relevant situation. And we went to a stressor that we were strongly believed is a chronic stressor, and that is keel bone fractures. Laying hens are very likely to sometime during their lifetime get a fracture of the keel bone, which is the bone that's sticking out in front of the chest where the breast muscles are attached to. So a little bit of debate where that fracture comes from, whether it comes from bad falls in these kind of multi-tier aviaries like this one here, or actually there's some evidence now that suggests that some of it comes from laying large eggs, and the eggs cause the tip of the keel bone to push out of the way and sometimes be fractured. The point is that by the end of life, usually many of the animals will have some degree of keel bone fracture. And we worked with a group in Switzerland under Mike Toscano's group, who had a study going where they took 11 x-rays through the life of the hens. So they really knew when the fractures happened in any given individual's life. At the end of life, we took a set of birds with minimal fractures because we couldn't find enough without any fractures, and a set of birds with quite severe fractures. And then we compared those to each other and found, so here we have two types of cells, multipolar and bipolar. They're both double cortin positive cells. They're just different stages of development. Multipolar are the more mature ones. And in both cases, we found that the group with high keelbone fractures, here in black, severe keelbone fractures, had many fewer neurons, double cortin positive neurons, than the control group, the minimal keelbone fracture group. And if you look, in addition to that, when we look at the multipolar cells, we also found that that effect was larger in the caudal hippocampus than the rostral hippocampus. So we found an interaction there between where in the hippocampus we're looking and the keelbone fractures. So that suggests again that the caudal part is more sensitive, and that, you know, adult hippocampal neurogenesis or double cortin densities respond to this stressful event of having keelbone fractures. In fact, when we looked at how long ago, so these are the times of the x-rays, first x-ray, second x-ray, third, fourth, fifth, etc. When the very first fracture was detected in an animal, we find that animals whose first fractures were detected very early on in the caudal hippocampus have fewer double cortin positive cells than those whose first fractures happened much later. So basically, animals that have been living with the fracture for longer have fewer double cortin positive neurons in the caudal hippocampus. Again, suggesting that it's sensitive to this duration of the stressor. So to summarize that, it looks like, you know, double cortin density is sensitive not just to having a stressor present, but also to how strong it is and how long that stressor is there, the duration that stressor is there. So we have a decent marker, it looks like, of negative experiences here. What we don't have, and I won't show you because we haven't done it, is done anything positive to see if the marker goes in the other direction. But now that we had this marker, we decided to actually see, so I've just summarized this here. So negative experiences, dose effect, but we don't have evidence for positive experiences. So we then decided to now flip it. If this is a marker of negative experiences, let's go test it against some things that we don't know if they're negative experiences or not. And so we started with housing, because everybody says, of course, free-range housing is high welfare, cages are low welfare. It's like, okay, let's see if that, you know, comes out with our marker of valence, potential marker of valence. So we collected animals from a local farm that had both colony cages. So these are large cages with different compartments for laying eggs and doing certain behaviors, but still, you know, they're cages with about 60 birds per cage. And they also had these multi-tier aviaries with pop holes for the birds to go outside. So we took birds from both of those systems to compare to each other. They were roughly the same aged birds. And because there's a lot of individual differences in there, we also collected from each system, we collected birds in good physical shape and birds in poor physical shape, because we wanted to control for the kind of within system variability that exists there. And because we can't process that many animals at a time, we didn't want to just randomly sample, because we might just have more good birds from one system than the other system. So we wanted to do it systematically. And so good physical condition birds means birds with a lot of feathers, with red combs, with good body mass, whereas we selected birds with pale combs, bad feathering, and quite skinny birds as our poor physical condition birds. So we went in, sampled those from the two systems from different places and different, you know, so they're not all from exactly the same location or anything. But we tried to sample the whole system. And again, to quickly cut to the chase on that one, we did not find an effective housing system at all, no difference. The main effect that you see here is really between rostral and caudal, which is not that interesting, this perspective. But within each, within rostral and within caudal, you could see that there's an overall effect of condition. The poor birds, poor condition birds, have fewer of these double cortin positive cells. And again, it seems to be a bigger effect in the caudal, but it's not significantly bigger. But we get fewer cells for poor birds than for good condition birds. So we don't find any evidence that, at least in this measure, that being in a cage versus a free-range system makes a difference to this marker, if we believe it's a valence marker. But what does make a difference is whether you are in good condition or you're a poor condition bird that might be pecked upon by the other birds in the shed. And so from a very practical perspective, it means that maybe just if we can survey how many of these poor condition birds there are in different systems, that might give us an idea for how good the welfare is generally in a particular system. And of course, it's easy to survey that because they're all external characters that you can check. Okay. Now, another one we were on a roll now. We said, okay, what about footpad dermatitis? I was at a UFAW meeting in Belgium a few years ago, and somebody was presenting about footpad dermatitis, and there was debate about whether this was a painful thing or not. And I thought, well, if it's painful, it's going to be chronically stressful. So why don't we compare birds with footpad dermatitis to birds without, see what that does to the hippocampal neurogenesis, tell us whether it's a chronic stressor or not. And so Matt, up here in the top corner, started his PhD, and within 2 weeks we had sent him to Belgium to go collect brains from these birds from a group in Belgium that just was coming to the end of an experiment and had birds with and without footpad dermatitis. So we took them, but unknown to us at the time, but we found out later, some of these birds came from a dark brooder system and some of them came from a conventional rearing system. Now, I'm telling you this because it becomes important for the rest of the story. Dark brooders are this system where the birds, the chicks, early in development, can hide under there to find warmth and darkness. And they can come out to go be active and then go back in to go sleep. Whereas in a conventional system, everybody's just in a big barn together, and animals that want to rest are just run over by animals that aren't resting. And so it's much... it's a very different way of being reared. And so there's a lot of arguments that being reared with dark brooders may be higher welfare. So that was part of the big experiment we collected the birds from, but didn't know at the time. But once we were told, we then did our analysis taking that into account. Now we're looking at Z-scores here again: caudal hippocampus, rostral hippocampus, bipolar cells, multipolar cells, conventional rearing here, dark brooder rearing here, and again here. Yeah. And then the controls and footpad dermatitis are the two colors. Now, we did not find an overall effect of footpad dermatitis. But to our surprise, we found an interaction between footpad dermatitis and rearing. And if you look at it, and it actually holds up in all 4 compartments, but I'm showing you in the one where it's the most obvious. In conventionally reared birds, we see what we expected. Footpad dermatitis reduces the number of double cortin positive cells in the caudal hippocampus. But in the birds reared with dark brooders, if anything, the effect went the other way around. Not significantly, but in the opposite direction. That made us really puzzled. Saying, what's going on here? One possibility is that dark brooders could be protecting against chronic stress or protecting against the experience of chronic stress. But of course, we have a chronic stressor, quote unquote, that we're not sure of is a chronic stressor, because that's why we started to begin with. We wanted to know if footpad dermatitis was a chronic stressor. So we can't say, well, it protects against chronic stress when we don't even know that footpad dermatitis is actually a chronic stressor. So we decided to do it experimentally. Go back to our unpredictable chronic mouth stress manipulation, and now do it in animals that had been reared with or without a dark brooder. And we worked with a local company in the UK who actually have commercial dark brooders. So we could take some of their animals that had been dark brooder reared and some that had not been dark brooder reared. And so we had 12 animals in each combination of stress and dark brooder, or stress not dark brooder, etc. Yeah. And we found the same thing. We found again in the caudal hippocampus and not in the rostral in this case, because it was a three-way interaction, we found that conventionally reared animals had a reduction in their double cortin. We actually measured gene expression here, but I don't have time to go into why that was. It was for very practical reasons of a freezer breaking down, essentially. But we measured gene expression, and again, we get lower double cortin for the stressed animals if they've been conventionally reared, but an increase in double cortin for the stressed animals if they've been dark brooder reared. So we get the same effect as we did for the footpad dermatitis, this time in a condition where we know it's chronically stressful because it's experimentally chronically stressful. So that puts us in a bit of a bind, right? If we think that this really is a valence marker, then chronic stress is enjoyed by the dark brooder reared animals, and that's fairly unlikely. So we have to think again about what does adult hippocampal neurogenesis actually mean. And so that made me think again about how stress and glucocorticoid levels relate to many things. For example, cognitive performance. And in many of those things, glucocorticoid or stress relates to the output measure in an inverted U type function. So many people have seen this. Stress, for example, for cognitive performance, a little bit of stress is good, but too much stress makes you fall apart. So what if neurogenesis does a similar thing? A little bit of stress or glucocorticoids increases it, but a lot of it decreases it. So if we had that situation, maybe our conventionally reared birds are here somewhere, with a decent amount of neurogenesis, and then we do a chronic stress manipulation and we push them along this, higher up the stress axis, and they end up with less neurogenesis. And in this interpretation, maybe we end up, here after dark brooder, we are, start with lower stress levels, and the increase in stress pushes them up the curve. So that's one interpretation. But we haven't got any good evidence, because we measured glucocorticoids, that they start in such different locations. Because for this particular system, you would have to start here for the conventionally reared animals and here for the dark brooder animals. So another interpretation of the same thing is that dark brooder rearing changes the reaction norm to stress. So if this is the conventional reaction norm and dark brooder rearing stretches that out, then if both of them start somewhere here, and then we induce chronic stress, that would decrease the neurogenesis for conventionally reared animals, but would increase it because of the wider reaction norm for the dark brooder reared animals. Now, this is where it's speculation, right? This is now me trying to make sense of the findings that we have. So if that is the case, then we're probably not talking about a marker of valence. But what is it a marker of then? And I know, I think I can do it. Well, basically what I'll do is I'll tell you rather than show you the evidence. There's good evidence now, in mice anyway, that adult hippocampal neurogenesis is causally related to stress resilience. Basically, you increase the number of new neurons, and in mice, of course, with all the genetic tools that exist, you can do this now. You can increase the number of new neurons experimentally, and the animals are more resilient to the experimental stressors that you give them, or you reduce the amount of adult hippocampal neurogenesis experimentally, and a smaller amount of stress will push them already over the edge, which normally would take more stress to do. So it's this study, but I'm not going to talk you through it. It's a really good paper, by the way. Annika et al. From Nature 2021. If you're really interested, I can send you the reference. But basically what they showed is what I just told you. So you can make animals more sensitive to stress by reducing either the activity or the number of new neurons in the ventral hippocampus, and you can make them more resilient to stress by increasing the activity or number of these new neurons in the hippocampus. So if that's the same in other animals, and we have no evidence for that whatsoever at the moment, right? We only know this for rats and mice, then maybe what we're looking at here is a measure of resilience rather than a measure of valence, where more neurogenesis means more resilient. And sometimes it will increase neurogenesis as a form of coping, but that it means higher resilience. Whereas if a stressor the stressful challenge increases neurogenesis, that basically means the animal is coping well and is activating its coping system or its resilience system. Whereas if stress reduces the number of new neurons, then basically we've pushed them over the edge. We are now beyond coping in a less resilient state. And of course, resilience is an important aspect of positive animal welfare. It's part of the definition that Brault et al. Came up with from the LIFT cost action last year. And so maybe what we have here is not a valence marker, but a resilience marker, which is still very useful in animal welfare, but is not the same as having a marker that says, I'm feeling good or I'm feeling bad. And I'll end there, only probably about just on time, to just thank the people that over the years have worked with me on this so far, several PhD students, technicians, and collaborators at different institutions and my own as well. And thank you very much all for listening. I hope this was interesting in my musings, make you think a little bit as well.
Yes, definitely, Dr. Smulders. Thank you for this presentation. I really like it, and I appreciate you sharing your expertise with us today. I'm sure our participants might have some questions. We can go ahead, and there is already one in the chat.
Do you want me to read it?
I can read it for you. So perhaps a question for later. So I will just read it. Running wheel exposure can also support conditioned taste aversion when novel flavor access is followed by running wheel access, albeit typically only when the running wheel is relatively novel.
Yeah, Tom, you're going to have to explain that.
Sorry, it was just in the context of you citing running wheels as a positive thing.
Yeah.
For rodents. Just that it might be more nuanced than that, given that there appear to be at least some negative aspects to it, particularly when the wheels are first exposed, so that there could potentially be situations in which unexpected results could come from the fact that the initial exposure to a wheel can create nausea, basically.
Yeah. Oh, well, the nausea. Yeah, I didn't even thought about the nausea. The nausea makes a lot of sense if you're in a running wheel. I did always wonder how they do it. Yeah, it's an interesting observation. We did a study where we gave the animals about 8 days of enrichment, including running wheels. And we found a very interesting difference that neurogenesis in the dorsal hippocampus went up, which would be kind of cognitive stimulation would be the typical interpretation of that. But it actually went down in the ventral hippocampus, suggesting that they might have been stressed by that if we interpret neurogenesis that way. And running wheels was a big part of that. So maybe the novelty of the whole thing is both stressful and exciting at the same time. And these things are not mutually exclusive necessarily. Thanks for the insight into the nausea induced by running wheels. That's one I hadn't thought about. That's cool.
Anna, do you have a question for us?
Yes, thank you. I'd like to make two sort of more philosophical reflections, and one is perhaps one I can imagine coming back to in more series here. I find it really interesting how you approach this valence questions and aiming to go in both directions, both positive and negative, which faces us with the question which you also reflected briefly on, what is an absolute neutral, and how do you know you're going positive rather than reducing negative? I'd like to ask you if you can develop that a little bit, and if you could perhaps, I at least find it useful to think about in human terms, because we're all humans, so we have some sort of shared experience here. What would be going positive versus reducing negative for as human academics?
I like the way that you put academics in there, because that puts it in a completely different sphere potentially than other human experience. I struggle with it myself, especially when I think about farm animals, when I think about lab animals, indeed wild animals. I think at heart, I'm a relativist. I think there is no such thing as an absolute neutral, even for humans. I think things are better than we're used to or worse than we're used to, but what we're used to is going to be individual to individual people and animals. I think it would be really good to get somebody from LIFT to come give a talk, maybe exactly on that issue, since positive animal welfare is the kind of core of that cost action. There's some people there who've thought about this a lot. And I still struggle with the, yes, that clearly more positive, but is it positive as opposed to less negative? And I don't feel I can give you a convincing answer because I haven't convinced myself yet how that exactly works.
Thank you. So here's an idea, Roish, for a future talk, and it's an appeal to LIFT members who are here. And Tom, if you can also perhaps offline later suggest names of people who could address this, because I think this would be perhaps not a topic of the talk of an entire talk, but at least something that would be really interesting and useful to explore more. Thank you.
Thank you, Anna, for the questions. We do have a lot of questions, but I am afraid that our meeting of this webinar will end exactly at 6, because this is the issue with the webinars. I might create it again and you guys might have to join it again. So let's take the questions and then I will update it, and within 2 minutes you can join again. So let's take our next question.
Shall I just go through the ones in the chat?
Yes, if you can.
Okay, so Liza's asking, what about the body condition? So yes, I think the implication would be that animals in better body condition are more resilient. Now, whether that's a direct cause and consequence, or whether that's just, animals that are in better body condition are in better body condition because they've experienced fewer stressors on them and therefore are more resilient still because they have not been battered down so much. But that would be the interpretation, yes, that the animals that we selected for their body condition, the better body condition ones, would be more resilient to stress. It could, of course, be the other way around, right? They might be in better body condition because they're more resilient to stress. And so that wasn't a correlational study, so there's no way we can tell the difference between those two without being able to do the manipulations people can do in mice. That's the challenge. The genetic manipulations in mice are very powerful, but not tools that are easily available for many other animals yet. Carol is asking about non-invasively measuring it, and as far as I know, still not. So as far as I know, we still need to actually kill the animals to take the measure, which has major consequences, of course, and makes the time course also much harder because you cannot do within subject studies. So Frank is asking about whether keel bone fractures would heal. And so one interpretation is that maybe it never heals completely and it stays annoying, if not painful, but it might be chronically painful. Again, we don't have no way of measuring that. I wouldn't want to rule it out just because there's no acute inflammation left, that there's not a long-term pain, because of course the animals are constantly moving with that. But again, open question. So the conditions are not related to housing conditions in the sense that we had poor and good condition birds, both from the cage systems and from the free range system. So in each we picked 12 good birds and 12 poor birds, and so therefore in our design anyway, it was completely orthogonal to the housing system. And so, yes, the interpretation would be that there's no difference for the resilience in birds in free-range conditions versus those in cages. And I know that's very controversial because free-range systems are supposed to be so much better for the welfare of the animals. But I think part of it, if we just go with human kind of perspective, is sometimes, as long as it's not absolutely bad for you, what you're used to is what you're used to. And so then the question is, we couldn't look, for example, at traditional battery cages because they're not legal anymore, so we couldn't find any of those in the UK. There are some places in Europe that still use them, but we had access to what we had access to. But my prediction would be that battery cages would reduce neurogenesis because they go below below what a normal animal could get used to. But that would just be my interpretation. And then about the inverted U curve. Yeah, that's an interesting question, right? Whether that would be other welfare measures might respond in an inverted U. I haven't thought enough yet to know which ones those would be. No, Frank, I don't think so, because valence and resilience are two different things. So positive welfare doesn't mean positive valence. Positive valence is an aspect of positive welfare, but not all of positive welfare. So I think they can be seen as two separate things. Although they're both contributors to positive welfare, positive valence of experience and resilience, which basically means not responding as strongly to negative things, are both aspects of positive welfare, but they're not the same aspect of positive welfare. So I do think they can be separated. And I think that's all the questions, Rohish.
Oh, yes, and on time, but we are still here, so I don't know.
It didn't kick us out.
Yeah, it didn't kick us out. But anyway, we learned that we should put 10 more minutes. But
Always a
thank you
good idea.
once again, Dr. Smulders.
I hope that was useful, yeah.
Yeah.
Yeah, I also like to thank you, Tom, not least for setting the spirit of these presentations as being a place where you can come with ideas that you're working on. This is all informative for all of us, and it's great to be listening to something which is really on the, I believe, on the forefront of what you're thinking about, because it's what you're working on.
It is.
And ideas you're working on.
Yeah, we need to get these data published. Thank you.
That was not what I was saying.
No, but you're right. It is data in that stage. Collected, analyzed, submitted as a PhD, not in a paper yet. Good.
Great.
Okay, before we close this, we should also add, Rohish, that we are planning for a next webinar in July. So we are planning for these to be bi-monthly. And Georgia Mason has agreed to give the next webinar. We don't have a title yet, but most of you know with Georgia and know that it will be well worth listening to. And we will disseminate this within a couple of weeks, the date and the title of the book.
Well, thank you, Anna. I will disseminate this information to the registered participants today and to our COST Action social media channels. So I once again want to thank you, Anna and Tom, and to all our participants. Thank you, and see you next time.